dear friends, welcome back. In this video, we'll be solving GATE-XL NAT questions of zoology paper. Let's begin. Question 92 of year 2024. The human genetic code is triplet in nature with 64 codons made using 4 nucleotides. If human genetic code was duplet in nature, the number of codons theoretically possible from 4 nucleotide is. Answer you should give an integer and expected answer is 16. As you know, human genetic code is triplet in nature. For example, if you take AUG, which codes for methionine, this codon, in place of A, there can be any 4 nucleotides, that is A, U, G, or C. And in place of U, there can be any 4 nucleotides, and in place of G, there can be any 4 nucleotides. So, 4 multiplied by 4 multiplied by 4, you'll get 64 codons. So the human genetic code is triplet. So therefore, there are 64 codons. If human genetic code is duplet in nature, for example, AU, in place of A, there can be any 4 nucleotides. In place of U, there can be any 4 nucleotides. So the number of codons will be 4 multiplied by 4 is equals to 16 codons. So if the human genetic code was duplet in nature, the number of codons will be 16 codons. Let's move to the next question. Question 101 of 2024. Consider the following nucleotide sequence. Here is the nucleotide sequence. Assume canonical initiation, canonical termination, no post-translation modification, and the average molecular mass of an amino acid to be 110 daltons. The theoretical molecular mass of the polypeptide translated from the above nucleotide sequence is. Answer you have to give in an integer, and the expected answer is to be in the range of 1540. Assume canonical initiation. Canonical termination means there is one initiation codon, that is start codon, and one termination codon, that is stop codon. And this polypeptide chain has not undergone any post translation modification. And they have given the average molecular mass of an amino acid, that is 110 daltons. So you have to find theoretical molecular mass of a polypeptide. So here they have given the nucleotide sequence. If you count the nucleotide sequence, that is, you will find 57 base pairs. So 57 base pairs this nucleotide sequence has. This base pairs has to be converted to number of amino acids. So to find number of amino acids, this 57 has to be divided by 3. Why we have to divide by 3? I'll tell you by using an example. AUG, this AUG, which is also a start codon, this codon codes for methionine. So this AUG, one codon has 3 nucleotides. So therefore, you have to divide by 3. So 57 base pairs divided by 3, you will get 19 amino acids. So this is the 19 amino acids polypeptide. Now in this 19 amino acids polypeptide, this has one start codon and one stop codon. So whenever you consider a polypeptide chain, you have to exclude this start codon and stop codon. So we'll remove this stop and start codon. So 19 minus 2 will give you 17 amino acids. So here is a 17 amino acids. So this polypeptide chain has 17 amino acids. Now to calculate the theoretical molecular mass of a polypeptide, you have to divide number of amino acids divided by molecular mass of amino acid. So divided by 110. This will give around 0.154. Now here what you have got, This is not an integer. So you have to convert this answer into an integer. So you have to shift this point to right hand side 4 times. 4 times means 0.154 multiplied by 4 times shifting, that would be 10,000. So multiplying this answer with 10,000 you will get 1540. The theoretical molecular mass of the polypeptide from the above nucleotide sequence is 1540 daltons. Thank you. This is the end of the video.